Okay, so I think we're live, right? So welcome everyone to the second design lecture of this MDDI cycle. We're thrilled to continue this series with a guest whose work bridges biology, art, and urban design. So today we welcome Dr. Elizabeth Henaff. Dr. Henaff is a computational biologist and artist with a background in computer science, plant biology, and bioinformatics. Her research explores the invisible world of urban microbes and the intricate way they shape and are shaped by our environments. And this has led to a diverse body of work from peer-reviewed research articles to high-profile art installations across, for example, New York City, Detroit, and Japan. And as an assistant professor at NYU Tandon School of Engineering, Dr. Henaff leads a research group that uses New York City as a living laboratory to study microbial metrics in urban spaces. And our team focuses on critical issues like green wall infrastructure, Superfund site remediation, and the impact of flooding on microbial diversity. And next to her research, she teaches biodesign courses and collaborates across NYU's many departments. So please join me in welcoming Dr. Elizabeth Henaff. Go for it.
Dan, thank you so much for the introduction. It's always great to be interacting with the MDDI community. So thank you so much for this invitation. So I know that y'all have just completed your bioregion module. And so what I prepared for you guys today is basically a brain dump about microbial metrics and how we can measure microbes in the environment. And I think that and why that's meaningful. And I think that it's particularly relevant for you guys to get at this particular moment in time because microorganisms are both hyper local, they're tiny, and the scale of their existence is extremely small. But also their impacts have global repercussions. So these are phenomena that span from the hyperlocal to the global. And so what I prepared for y'all today is kind of a crash course in what is a microbiome, how do we measure it, and how do we design for it. So there we go. In the sciences, oftentimes, we study things that we can't see, that we can't perceive with our direct sensory system. And in order to do that, we need to build tools or instruments to be able to extend our sensory system. And so here on the left, for example, you can see an image. This is a scanning electron microscope image of condensed chromosomes poised for cell division. And so the microscope, for example, is a tool that allows us to see the very small and to understand things like structures of chromosome and other cellular functions, for example. The image that we see in the middle here is an X-ray. So that's a technology that allows us to see the bony structure of a living animal without the need for dissection. And so that gives us insights into physiology and biomechanics, for example. And the image on the right we see here is an example of a DNA sequence. So DNA sequencing can also be considered a tool that allows us to get a digital insight or a digital readout into the molecular basis of the identity of a living organism. So even though DNA sequencing is more like a process than a tool because we go through multiple steps, we can imagine it as this tool that we can focus on different living organisms to be able to understand them from a genetic perspective. And so that tool has been extensively used in the context of human medicine. So precision medicine is kind of a field of medicine that has emerged as a function of the availability of this technology, where, say, if a patient is diagnosed with a cancer, the oncologist will prescribe a whole genome sequencing of the cancerous tissue and healthy tissue, and then being able to look at those genetic differences can provide insights into treatment plans. So we can use that tool to face it upon ourselves as human organisms to understand our functioning better, but we can also use that tool to understand organisms in our environment. And so if we think of the environment, we might think of plants and animals and lush green landscapes as represented in this still here from one of my favorite all-time animes, Princess Mononoke by Studio Ghibli. But oftentimes what doesn't make its way into our imagination of the environment are microorganisms. So if we look at this, fictitious image here, what we're not seeing represented would be the billions of microorganisms in even just a little teaspoon of soil from this forest landscape. And DNA sequencing is a tool that enables us to perceive those organisms without the need of culturing them. So without the need of putting them in a petri dish and seeing if they're going to grow in a lab. This is really meaningful because a lot of microorganisms do not like to grow in a petri dish. And so that particular tool presents a certain bias because many organisms, we call them recalcitrant microorganisms when they don't want to grow in the lab. So this particular tool of DNA sequencing gives us a less biased insight to the microbial component of our environments. And this is meaningful because there's a whole body of literature that's emerging that shows that physical characteristics or phenotypes of multicellular organisms that were thought to be strictly genetically controlled are actually modulated by the nature of the microorganisms with which they interact. So some examples. It's been shown in plants that you can alter the flowering time of plants by altering the composition of the microbiome of the soil in which they're growing. This was a really big deal in the plant biology world because flowering time was thought to be the canonically well understood, genetically determined pathway. So different plants have different strategies for reproduction, integrating different types of environmental factors like temperature and light. And it was thought to be strictly genetically controlled by the genetic makeup of the plant itself. So this is an example where a phenotype or physical characteristic of a multicellular organism is determined not only by the genotype or the genes of that particular organism, but also the genotype of the microorganisms with which it's interacting. That's been described in plants. It's been described in mammals as well, including humans. And so we're we're familiar with the idea of the gut microbiome in humans being important for gut health, for example. But the gut microbiome also modulates different, more subtle kinds of phenotypes in human organisms. So, for example, the gut microbiome will determine will regulate the synthesis of different neurotransmitters in the brain. So affecting things like mood and things like that. So this is just a couple of examples of a whole wide body of literature that's emerging that shows that physical characteristics or phenotypes of multicellular organisms like plants, mammals, humans are due not only to the genotype of that organism, but also the genotype or the nature of the microorganisms with which it's interacting with in the environment. And so that kind of begs a redefinition of the notion of the individual to encompass not only the host's genome, but also the genome of its symbionts. And so the word that has been coined quite a long time ago to describe this kind of broadening of the notion of the individual is holobiont. So this term was coined in the 70s before the advent of DNA sequencing technology being as extensive as it is now. But so now we can talk not only about the genome of a person, but the idea of the hologenome. So the genome of the holobiont, kind of broadening this notion of an identity. And so this is meaningful for design of urban spaces because urban spaces are thought to be designed for humans. But if we kind of accept the fact that human beings have porous boundaries and their health and well-being is tightly linked with the types of microorganisms that they interact with, so more with their holobiont, we should be designing instead for the human holobiont and not just for the human being. So if we imagine our urban environment here, we have our built environment, structure, man-made materials, and then the humans that are occupying it. But if we were be able to point this kind of, let's imagine it at this instrument of DNA sequencing to this environment, we would be able to see all the microbes in this environment. So that would be the human-associated microbes, that would be the environmental associated microbes. And what's meaningful here is that kind of human health and well-being is tightly related to our relationship with our microbiome, but also our microbiome is interacting with and impacted by the environmental microbiome. And the environmental microbiome is sculpted by design decisions made by design practitioners. So architects, city planners, material scientists. So the nature of the materials and the shapes here of our built environments are impacting this environmental microbiome, which we kind of could think as a separate thing, but actually it's a continuum between the environmental microbiome and the human microbiome. So if we think of principles of design in the built environment, in general, we have choices between two main things. We can choose materials and we can choose shapes. And most of the time, these choices are optimized for human comfort. Here's a couple of examples. We can think of thermal comfort, for example. So, optimal, comfortable temperature for a human being in an indoor environment might be like 19 degrees Celsius. And this image here of like stucco construction from the American Southwest is an example of choice of materials and shapes to optimize human comfort. So these buildings are built with extremely thick walls, with small windows and apertures to optimize human comfort in an extremely arid and hot climate. We can also think about acoustic comfort, for example. And this example is a concert hall in which the materials and in particular, the shapes of this concert hall were optimized in order for the best like acoustic comfort and experience of the listeners in this concert hall. So this is just a couple of examples, but I'm sure you have many examples in your own design practices of kind of these choices between material and shape to optimize a particular metric. And so the idea of designing for microbes is not very much different. We still have choices between materials and shapes, but here the metric that we want to optimize for is optimizing for human microbe collaboration. And we're going to talk a little bit more about what that means exactly. And so the idea here is that what we want to be able to do is kind of optimize this environmental microbiome so that this environmental microbiome is the kind of microbiome that we as human beings want to be interacting with in our built environments. And so to be able to do that, we need to be able to do a bunch of different things. We need to be able to first be able to measure these microbiomes accurately. We need to be able to make accurate maps. We want to be able to represent them in meaningful ways at the scales that are meaningful for us, which is actually quite tricky because microbes exist at a microscopic scale. And when we're designing for the built environment, we're designing at a human scale. We might want to alter the microbiomes and their functions. We certainly want to design for them. So to kind of encourage the microbial populations to go in a particular direction from maybe a metabolic perspective. We want tools to be able to interact with them, to be able to get feedback from those environments and also provide feedback to them. And so I wanted to give you just a few examples of tools that we have for each one of those different steps. So tools that we have to measure microbiomes, currently the most precise ones are based on DNA sequencing technologies. You can do that in the lab, but you can also do that in the fields. This is an example of a field-ready DNA sequencing device that you can plug in here to the USB port of your computer, and with really minimal preparation, you can take samples from the environment and get a DNA sequencing readout immediately. This has been used in biosurveillance contexts and also in the field. You need computational algorithms to then interpret that DNA sequencing data that you're obtaining. So you go from your kind of mixed bag of microbes to a mixed bag of unknown DNA sequences, and then you need computational algorithms to be able to make sense of those, map them to known databases of known microbes to be able to identify, for example, different taxa or different species or different metabolisms. You need to be able to represent this data at the scale that is meaningful for you in design. So this study is almost 10 years ago now, but this was the first study characterizing the microbiome of a transportation system. So this was the subway system in New York City. And this was a really awesome interactive map that the Wall Street Journal made in response to this study. And so if you happened to be in New York at that time, you can click through that and see what kind of microbes you're interacting with on your daily commute. Making microbial maps is not an entirely new thing. This is a map made by Dr. John Snow in the mid-1850s in London during the cholera outbreak. And so what he did was color in a dark color the houses that were affected by the disease. And so by using this data visualization, he was able to identify that what those homes had in common was the use of a particular well, and identified that as the source of contamination. We want to be able to design for these microbes that we're measuring in different spaces. So this is an example of a material that has been optimized to host microbial growth in urban environments. And so this material is bioreceptive cement. It was developed at a lab at the Polytechnic University in Barcelona, actually. So this is an example of a building material that is designed not only for its structural characteristics, so it needs to be able to, have certain compression strength and things like that, but also for its microbial characteristics. We also need to be able to interact with this invisible living component of our environment. And so we have tools to collect biomass from the environment. So we see here, this is a air quality monitor here in the subway in New York. And we're all very familiar with the ubiquitous swab, right? That we got way too comfortable with during the years of COVID, of having this stuck up our nose for COVID tests. But this is the most common tool used to collect biomass from our environments. These tools don't feel super comfortable, right? Like this feels like some kind of surveillance apparatus. The swab here makes us think of disease and contamination. And so we're going to talk a little bit later about how the design of the tool really matters when we're interacting with something that we can't see, because ultimately the interaction is going to be with the tool more so than with that material. So, we have tools to monitor pathogenic microbes like we just saw, but we also have means of interaction to supplement the human microbiome. So we're familiar with, taking probiotics for gut microbiome, but there's also a whole field of the, I guess, beauty and wellness industry that is looking at probiotics in other formats. So this is actually the very first company making probiotic skincare. So if we put all these together, if we have good ways to measure things, good ways to make maps, tools to be able to design for microbes, and interfaces to be able to interact with them, then we can start integrating these microbial metrics into designs either at the city scale or the human scale, the city scale, or also the region scale. One of the real challenges is the fact that these guys are tiny and that we can't see them. So if we were able to if I had a superpower, my superpower would be to be able to see microbes in the environment. I think that would be awesome. And if you guys are interested, this is a really awesome manga now turned anime called Moyashimon, that is the story of a kid at an agricultural school who's able to see microbes in his environment. And if you want some fun binge watching that you can call studying for the next few weeks, I suggest that you watch this anime. It has a lot of really like culturally relevant questions that they raise. But just to give you an idea of scale, of small, when we think about, oftentimes we kind of lump in the same category anything that's too small for us to see, but there's actually vast orders of magnitude of small within the invisible. So if we think of the scale of the human body being measured on the meter scale, human cells are measured on the tens or hundreds of micrometers. So here we're talking even a million times smaller. And then bacterial cells are orders of magnitude yet smaller than a human cell. And then the width of a DNA molecule is orders of magnitude yet smaller than that. So if you think of kind of an analogy, for example, so a bacterial cell is about 100 to 1,000 times smaller than a human cell. And so this is kind of some like brain stretching math to do, but ultimately in the human body, we have pretty much the same number of microbial cells as we do human cells. But the reason why we don't feel like giant bags of walking microbes is that those microbial cells are so much smaller. They're hundreds or thousands of times smaller than that. But say if the cells in your body were to vote about something, your microbial vote would weigh almost just as much as your human cell vote. So with that kind of in mind, kind of with this structure of scale and size, I want to give you guys a crash course in the biology of microbes in the environment. And so we're going to go through real quick microbial cell structure, how they're different from human cells or mammalian cells, talk about microbial community structure. You know, microbes are unicellular organisms, but they also very much exist in communities. And then I'm going to give you some kind of literature review about microbes in soils and plants and mammals and the built environment. So I'm going to go really quick over this. The idea just is to provide you with some further reading should you be interested in any of these topics. So the microbial cell or prokaryotic cell that we see here on the left of this image is quite different from an animal or eukaryotic cell. So it's different in a couple of significant ways. First of all, it is a good bit smaller, and kind of a corollary to that is that it is an unstructured cellular environment. So the animal cell has a bunch of kind of pockets or cellular substructures that you can see here that perform different functions. So the nucleus that stores DNA, for example, the mitochondria that generate electricity or energy, but also, yeah, kind of electricity. And so this compartmentalization of a eukaryotic cell is what allows it to get so much bigger than a prokaryotic cell. So if you've ever wondered, like, gosh, my gut microbes are 1,000 times smaller than a human cell, why is it that they're not getting bigger? This is the reason they're limited in size. So we've talked a little bit about DNA and how DNA sequencing is an insight into an organism's identity. And so an analogy that's helpful to understand that is that we can think of DNA as like the relationship between DNA to a cell structure is analogous to the relationship between a blueprint and a construction site. So a blueprint holds all of the information necessary to build a building, but it is not the building. What it is it holds the kind of instructions for the machinery in the construction site to assemble materials in the right kind of order to be able to build a building. And so the analogy here for the cell is that the DNA is similar to the blueprint, and what it gives is instructions to the machinery in the cell, which would be enzymes, to assemble materials in the right order. So forming things like cell membranes, for example, or synthesizing other proteins that are necessary for the structure or the function of the cell. So hopefully that's a useful analogy as you think about the difference between the information storage, which is the role of a DNA molecule, and structure or function, which is the role of a protein in a cell or enzyme. Microbes exist in communities. So just thinking here about the difference in cell size, we have here our animal cell and our prokaryotic cell, and this is a microscopy image that shows the difference in size. So this big lump here is an animal cell, and these small lumps here, which are actually kind of assemblies of multiple small cells, are bacterial cells. So you can see the difference in size here. Microbes are unicellular organisms, so we, define an individual in among microbial species as one single cell. It is a kind of self-sufficient unit most of the time, but microbes do exist in communities, and those communities have very specific structures. So usually we call microbial communities biofilms. Those two words are often synonymous. And microbes have the ability to sense their place in a biofilm and take action as a function of that. We call that quorum sensing. So they're able to sense the nature and diversity and composition of microbes in their environment, in that biofilm, and they can make decisions according to that. Do I stay here or do I leave here? Do we dissolve the biofilm because we've used up all of our resources, and do we go somewhere else? So microbes are very much aware of their environment, but not only the chemical nature of their environment, but also the living kind of community nature of their environment. These are some cool pictures of biofilms that can show the diversity of microbes in a biofilm, but also the very precise three-dimensional structure. So what we're looking at here are microscopy images where different microbial species have been tagged with different colors of fluorescent molecules. So each color here represents a different species, and the biofilm that we're looking at here is tooth plaque. So the biofilm that forms in between teeth. And so what we can see here is that these communities are very well organized. So if we look at this picture here, bottom left, you can see that there's green species here kind of on the outside of this three-dimensional structure, and filaments of purple species here on the inside. And so of what we can note here as far as microbial cooperation is that the purple species here is anaerobic, so it wants an environment devoid of oxygen, and the green species here on the outside is aerobic. So what's happening here is that there's a three-dimensional assembly of these two species in which the green species is creating an environment and is consuming all the oxygen, creating an environment for the purple species to be able to survive. So we definitely see cooperation in the assembly of microbial communities. Lichen is always a cool example. You can find it everywhere, anywhere where, you on rocks and things like that. And lichen is interesting because it's a collaboration between microbes and an organism, not even in the microbial kingdom. So usually it's a collaboration between a fungus or an algae. And so these are very ancient beings. They're one of the first forms of kind of multicellular collaboration. And you can find them everywhere. And so what's interesting about lichen, which I really appreciate, is that the superstructures that they form, like the biofilms that they form, are visible to the naked eye. And so that's one example of how we can actually just see microorganisms in our environment without the need of a tool that mediates that relationship. We find microbes in all environments that have been studied to date, independently how extreme or how harsh those environments might be. So we find microbes in soil. Microbes adapt very quickly to soil conditions and anthropogenic contamination as well. And so this is a cool paper that shows that you can use the composition of microbial community as a biosensor to understand aspects about that environment, kind of abiotic aspects of that environment itself. Microbes adapt very quickly to anthropogenic contamination. So this is a great paper that shows how microbes responded very quickly to an oil spill on the Gulf Coast in the United States. Microbes coexist with plants. So there's really beautiful collaborations between plants and the root structures of plants and microbial species. So rhizosphere is a word that you can use to find out more about the kind of three-dimensional structure that is created by plant roots in soils and how that hosts microbial species. So in particular, legumes, so plants in the general family of legumes, including soy and clover, for example, have very explicit collaborations with microbes in which they will create these nodules that house microbial species that will fix nitrogen from the soil. And so there's a symbiotic relationship here. And from a design perspective, you can see how these plants have evolved to create three-dimensional structures that house these microbial species. Understanding microbes in the soil is important also for agricultural activities. So microbes in the soil are what are mainly responsible for the fermentation and flavor profile in naturally fermented wines. And also a lot of the plants that we use for medicinal reasons, those secondary metabolites that are the meaningful molecules that for which we harvest those plants, so in some cases, those secondary metabolites are produced by the microbes that live in and on the leaf structures, not the plants themselves. We've talked about microbes in mammals a little bit, especially humans, but this is a really interesting paper that gives kind of a survey of how microbes interact with mammalian development in all phases of the development cycle. So from fertilization, embryogenesis, etc., to adult phases. Interestingly, the human microbiome is maternally inherited, so it's a matrilineal characteristic. We talked about microbes and the brain, and I'm just going to leave those for y'all to read. So microbes in the built environment is a kind of new field, but there's an emerging body of literature that is describing that. We talked about the subway paper. This is another paper describing microbes in the built environment or in transportation systems. And that was really interesting because they related different microbial composition, like community composition, with different materials within the transportation system. So the microbiome of the plastic handle was different from the microbiome of the steel pole, which was different from that of the fabric seats. And so this is an example about how material choices, which is something that is very important in the design process, can directly influence or select for specific kinds of microbiomes. This paper describes how the design structure of like indoor environments impacts the microbiome of these indoor environments. So this is talking about connectivity of indoor spaces. And so how the connectivity of different rooms in a building will impact how the microbiome is structured on the inside of this building. Microbiomes are great in many ways in the built environment, especially with respect to, diversity with respect to human health, but they can also have very negative impacts on our materials, the materials that we use in our built environment. And so as kind of a word of caution in designing for microbes, we also need to be careful because these microbes are also able to degrade a lot of the materials that we use in the built environment. And we talked about bioreceptive concrete. How am I doing on time? Okay, I've got 15 more minutes. That's good. So we've talked about how microbes are basically ubiquitous in any kind of environment that we might want to study. And so for that reason, that's why I believe that it's important to incorporate microbial metrics in any kind of design that you guys might be thinking of, because they're going to exist within those things that you design, whether you like it or not. And so being able to kind of make those designs explicit for microbial metrics is very important because of their impacts on human health. So we've kind of done kind of a 10,000 foot overview of all these different tools that we need to set up to be able to design for microorganisms. And what I want to talk about now is why the design of the tool itself is important. And it's important because we're not able to see microbes in the environment. And so the relationship that we have is with the tools that we have to be able to perceive those more so than with the organisms themselves. And this is meaningful because the tools are going to mediate different kinds of relationships depending on their design. So if we see the swab here on the left, it's giving like contamination, sickness, hospital environment for perfectly good reasons, because it is a clinical tool designed to be used in a clinical environment. So when we were doing that subway study, and there was like groups of undergrads running around the subway taking samples, there was a lot of really strong reactions from people in the public who were witnessing that. And so people were concerned that there was some kind of epidemic that we were monitoring for. This was pre-COVID too. This was 2015. One group also got accused of implanting HIV in the subway. So people were concerned about bioterrorism. And what the reaction that they were having was a reaction to the tool itself. It wasn't necessarily a reaction to the fact that we were interested in studying microbes. But if you see the image on the right here, like grabbing a handful of soil is also collecting a microbial sample, because there's going to be just as many microbes in that handful of soil as what you swab with your swab, but you have a very different reaction to that action, right? Like grabbing a handful of soil feels grounded and connected and earthy, and you can have all sorts of like positive words to describe that. But ultimately, these two things are doing exactly the same thing. And so that's why the design of interfaces really matters when we're interfacing with microbes, because the interface is what's going to dictate the relationship. And that's why I think that as designers, your role is very important, because you are kind of directing the valence or the positivity or negativity of the relationship in designing the tools to interact with them. And so, unfortunately, the tools that we have available to us very much fit in this kind of clinical biosurveillance realm. And so I think that the work to do now is to think about designing tools for more positive interactions. If you're interested in thinking more about kind of the philosophy of science behind this idea of instrumentation and the bias in instrumentation, I strongly encourage you to read work by Karen Barad. She's a physicist and a feminist theorist, and has written a lot in the context of philosophy of science and feminist theory about how the tools that we make for scientific research are not these kind of unbiased extensions of our sensory system, but very much carry the inherent biases and wishes and desires of the person who is making that tool. So she uses the word apparatus as a kind of synonym for tool or for instrument. And so notably, she says that apparatuses are themselves phenomena. They are actions. They're not just passive. So I encourage that as some reading. What this means for me personally is manifests in art practice in addition to the publication of scientific papers. So I've, published several papers, developing instruments to be able to sample biomass from indoor environments or urban environments. But what's meaningful to me to be able to think through how we can design interfaces for meaningful microbial interaction comes in art practice. So this was a show that was produced at the Storefront for Art and Architecture in New York City, where we were commissioned to redesign the facade of the gallery with the imagination of what would it look like if the built environment were designed for microbes instead of for human beings. And so here we designed a bioreceptive wood facade for the facade of this gallery. What it also means for me is using technology to be able to adjust the scale of human observation to be able to understand microbial systems. And so this is a set of stills of time-lapse photography of an aquarium that we set up in the lab with contaminated sediment collected from a contaminated waterway here in New York City. And so what we see is that, at first observation, it looks like there's just nothing happening there. It's just kind of this dark black sediment. And then if you give it enough time to do its thing, you can actually observe that it's a quite dynamic ecosystem. So here we see a big algae bloom, and then there's these microbial mats or kind of biofilms that form. And then there's algae that overtakes the aquarium. And so what was meaningful to me in this experience is that whenever I would go to the lab and I would look at the aquarium, I'd be like, ugh, nothing's happening. But then having this tool of, time-lapse photography allowed me to be able to sense this at a time scale that was more meaningful for microbes and that was very different than the time scale that's meaningful for us human beings. I've collaborated with a really awesome group of folks we call ourselves the Scope Collective to produce art exhibits to try to make apparent these like dynamic characteristics of invisible microbiomes. This is a show that's currently up at a gallery in New York called Biobat with some giant tanks and also projected imagery. This was a previous show also here in New York City, really like based on photographic imagery, like high-res photographic imagery taken of microbial structures that we were growing in the lab and the built environment, with the idea of kind of inverting this power dynamic related to size between microbes and humans. So trying to make the microbes very large and kind of large scaled compared to the human scale. And so in thinking about tools and designing interfaces for microbes, it's easy to imagine our understanding of the microbial world as something that is specific to our kind of modern understanding of biotechnology. So we've had access to DNA sequencing for, the last 20 years. The human genome was published in 2001, I believe. So it's kind of easy to imagine this relationship to microbes as being a new thing that we're understanding with the tools of like modern Western biotechnology. But we have never not lived in a microbial world. So microbes existed for billions of years before multicellular organisms ever developed. And so human beings have always existed in the context of a microbial world, and we have always interacted with microbes for really meaningful ways with respect to our life ways and survival and nutrition. And so I think it's important to keep that in mind, is that there are cutting edge recent biotechnologies that we can think of, that we can design, that we can imagine, that allow us to interact with microbes. But there are also long-standing ancient technologies that also mediate those relationships. So for example, fermentation is an easy one to imagine. We've been using fermentation for food preservation for thousands of years. And that practice has evolved in pretty much any place in the world that you can study. So this is an image from a book that was imagined to be published in the 1300s. But the oldest bioarchaeological record that has been described of food fermentation was dated to 9,000 years before the current era, and is a set of holes dug in the rocks in what is now Northern Europe that was determined to be used for fish fermentation, for preservation. So there exists very ancient technologies that allow us to interact with microbes. This is an example of another ancient technology in which rain dances, which are rituals practiced by indigenous populations all over the world, are hypothesized to work through the phenomenon of bioprecipitation. So precipitation or clouds form in the atmosphere when there's moisture and also some kind of nucleating agent. So particles in the atmosphere like dust or ash, where the water molecules in the atmosphere will form crystals around these nucleating particles, and those crystals are clouds, and then clouds leads to precipitation. And so there's a hypothesis that rain dances work through this phenomenon of bioprecipitation because there's a particular microbe named Pseudomonas syringae, which is a plant pathogen. So it lives on plant leaves. And that microbe produces a protein that acts as a much better nucleating agent than non-biological particles in the air. And so there's this hypothesis that this ritual practice mediates like an interaction or an interface with the microbial world, specifically for the purpose of precipitation. And so speaking of aerobiomes, I have kind of a call to action for you guys. So we're, now very aware of air being a fluid medium that carries biological particles after years of COVID and thinking about indoor environments and contamination and things like that. And so it feels kind of like new, I guess, to us to imagine the air like, carrying biological particles. But COVID is certainly not the only thing that air as a fluid medium carries. And bioaerosols are actually a well-studied field, and we have a whole bunch of different types of biological particles in the air that mediate really important weather-related or climate-related phenomena. And so microbes have been described in like the air as a fluid medium as a planetary phenomenon. So there's microbes in the air that come from different soils that also come from bodies of water. And if you can imagine, and they have really fundamental roles in weather patterns and climate. So bioprecipitation is something that I encourage y'all to read about. And again, this feels like maybe a new thing, but this is a really awesome book that describes living organisms in the atmosphere that was published in the late 1800s and has some cool diagrams of instruments that people have made to study microbes in the air. But I think what's really important is thinking about this from a policy perspective. So this is the call to action, is to, I guess, realize, as you are doing your regional studies, that the biology of each one of our regions does impact planetary scale phenomena such as climate and weather, and a lot of that impact is mediated by the transfer of biological material and microbes and other particles from one region to another, and its impact on precipitation. And so I'll leave, so this is a map that I like to refer to. It's a really fun website to look at. It's called windfinder.com, and it shows you global wind patterns like in real time. And so you can see that these global wind patterns, you can imagine them transporting biological material from one continent to another. And that has been well described. Like we find microbes from the Sahara Desert in Brazil that are carried there by the trade winds between the two continents. And finally, what I would like to, my second call to action is that a lot of the work that I've described here and that I think is important to do in the context of design and biology is interdisciplinary. And what that means is that interdisciplinary work leads to diversity of outcomes. So maybe the work that you're going to be doing can be published in a paper, but also maybe it's meaningful to make an art exhibit, or maybe it's meaningful to write from a philosophical standpoint about this work. And one of the challenges with interdisciplinary work is the fact that these outcomes tend to be siloed in different kinds of forms and siloed with respect to different communities. So a scientist isn't necessarily going to go see an art show, or an artist isn't necessarily going to read a scientific paper or a philosophical paper. And so what I think is really meaningful is finding avenues where these interdisciplinary outcomes can, or this interdisciplinary work can be published in outcomes that are meaningful for different communities. And so particularly relevant to you guys, what I wanted to share is that there's this new journal from Cambridge University Press called Biotechnology Design that aims to house publications that exist at the interface between scientific, like biology disciplines, and also arts and design. It is organized around research questions. So there's probably a dozen of them. A few of them are here. So one research question here is, how do we design with materials that have their own agency? Biofutures for transplanetary habitats, living textiles. So if you're working at the interface between biology and design, I encourage you to check it out. If you have any questions, I'm on the editorial board and I would be glad to answer them. The question that I'm championing, that I'm particularly attached to, comes back to this idea that biotechnology does not necessarily belong only to Western science. And that vernacular biotechnologies, so for example, objects, that are designed to interface with a living component of everyday life or the environment, can be informed by traditional ecological knowledge and also performed by people who do not necessarily identify as engineers, and also taking place in places outside of the lab. So biotechnology also happens in the kitchen or in the field on a farm. And so this particular research question invites contributions in that vein. And with that, that was everything that I had for you guys. I thank you for your time and attention. And I'm glad to take any questions.
Thank you very much, Elizabeth, for this verbal presentation. Yeah, if there's any questions from the students, please open your mics and ask them. And I will keep also an eye on the chat on YouTube. If there's any questions from our online audience, please ask them there, and I will refer them. Go for it.
Hi. Yep, I have a question. Thank you, Elizabeth, for this amazing presentation. I'm particularly interested in what you mentioned. The room's connectivity also impacts, their behavior. So I'm wondering, it's sort of similar to how spatialized sound is functioning and also researched. I'm wondering, is wave is considered in the studies as well?
Oh, are waves like do people use waves to model that? That's an interesting question. I think yes, to the extent that a lot of the modeling that's meaningful for that is modeling of airflow, airflow and also temperature gradients. So usually in indoor environments, the microbes that exist in indoor environments are microbes that are brought in from the outside. So either brought in by human inhabitants and the microbes that we're shedding all the time, or also microbes that are related to inputs from outdoor air. So depending on the nature of the HVAC system, that can mean different things. So if it's a building like a home without centralized air, then diversity is usually going to be related to the distance from a window that can be opened. But if there's a centralized HVAC system, then those diversity metrics are going to look different because the air circulation and airflow is going to look different.
Yeah, it's interesting. It's about the movement. I'm wondering what about the geometry, for example, of the room. It seems like a super mega structure for, microbes.
Yeah, certainly. So they have some really nice comparisons between and they kind of define like a geographical metric in a certain sense to describe connectivity of a given room space. So like a highly connected room would be a room, say, that has doors on all four sides and that connects to multiple other spaces. Or a low connected room would be a room, say, at the end of a hallway that only has one entrance point and exit point. And so they're able to relate that metric of connectivity of spaces to diversity in microbiomes.
I see. Yeah, that's amazing. Thank you.
You're welcome.
I have a question from the comments here. An architect saying, thank you for the great presentation. And I wanted to ask, what is biosurveillance?
Biosurveillance? Oh, yeah. So biosurveillance refers to the process of monitoring the environment for biological agents. Usually it's targeted towards pathogens. So being able to monitor an environment for particular known pathogens. And so usually that's implemented with targeted approaches. So you have a list of pathogens that you want to be able to get a yes or no answer, like is it present in this environment or not present in that environment? And so usually you use targeted strategies to be able to identify whether that pathogen is present or not. Obviously, I'm interested obviously, I'm interested in characterizing microbes in the environment from a more holistic perspective. So not just answering the question, is there a pathogen there or not, but asking the question, what is the entire population of microbes in an environment? And those two approaches relate to two completely different goals too, because usually biosurveillance is related to the goal of eliminating pathogens from an environment, while characterizing the diversity of microbes in an environment is usually related to the goal of promoting diversity. So promoting a diverse, like, microbial population in an environment. So those usually have two different goals.
Awesome.
I also have it made me think of some sustainable building. They will also use plants on the facade of buildings, even indoor. Some people say that will impact human skin, and if we use airflow, how much energy do we need for that? And can microbes also contribute to the sustainable energy? Maybe doing chemistry with sunshine, etc.
Yeah, I think that green wall infrastructure indoors and outdoors tend to have very different goals. So you're right. So green wall infrastructure on the outside of a building usually corresponds with goals of energy reduction. So, because the plants provide shading, for example. And on the indoors, besides the obvious impact of biophilia, because we like seeing plants in our environments and it makes us feel good, you can also design those green walls with the goal in mind of inoculating indoor spaces with microorganisms. There's a rock star PhD student who just got her PhD who has worked with my lab, who's at the Yale School of Architecture, who has done a lot of work around that. And so what she found is that if you're designing green walls with the goal of inoculating indoor spaces with microbes, what really matters is the growth substrate in which those plants are growing, with kind of a spectrum between hydroponic substrates, which have very low microbial diversity, and soil substrates, which have very high microbial diversity. So it's not just the plants that are hosting the microbes, but it's mostly the soil that they're growing in that are hosting it. So there's kind of two major design criteria that you can take into consideration, one being the substrate, and then the other one, like you mentioned, being the design of the airflow through that green wall.
I see. That's interesting. What about the electricity carried in the cells? Do they count in some, studies? I'm curious.
Oh, like using kind of like the biochemistry of microbes in the soil to generate electricity?
Yeah, or more specifically, I'm currently doing some research in oyster, and yeah, it would be interesting to think about microbes in the oyster, and I'm wondering, can they generate the electricity as small infrastructure for, as small devices?
That is not my field of expertise. I don't think I can answer that question.
Oh, why not? Because the oyster?
Yeah, I'm not familiar with oyster biology and how
That's
they
fine.
interact with microbes. I mean, I know that they are very sensitive to water quality,
Yeah.
and have been used as biosensors for water quality, but I imagine that that's an integrative metric, and so they're sensitive to kind of a combination of both chemical contaminants in water as well as biological contaminants.
Okay, cool. Thank you.
Yeah, you're welcome.
I have another question from the comments. Is there a target group of microbes that you want to have, for example, for indoor human health, or is it more a question of diversity?
It's the latter. It's difficult to be prescriptive about what species one would want in an indoor environment, for the reason that everybody is going to be different. So everybody's gut microbiome is going to be different, for example. So an environmental microbiome, what would be an optimal environmental microbiome for one person or another is likely going to be slightly different. Similarly to the gut microbiome. So there's been studies that, look at gut microbiome composition across a bunch of healthy individuals. And when you look at it from a species perspective, they look wildly different. So there's completely different types of species and abundances. When you look at them from a functional perspective, so like what kind of metabolisms do they perform, they are very, very similar. So when you're thinking about designing microbiomes for human health, I think it's more meaningful to think about it from a functional perspective. So what kind of metabolisms do they encode? And it's actually fine if it's different players that are encoding those metabolisms, but what's meaningful is the metabolisms themselves. But generally, if one is looking for like a quantitative metric to optimize for, it would certainly be diversity.
Great. And another question from one of our MDDI alumni, Annie. I don't know if you remember her.
Oh, yeah.
For material design perspective, what do you think about biomaterials? Is it preferable to have natural or artificial, or it doesn't matter at a microbiome perspective? I
Oh,
hope
from
I
a
got it.
bioreceptivity standpoint, I think that in general, biomaterials are often based on some kind of structure that was very much bioreceptive when it was alive. And so it's more likely for it to be bioreceptive once it's been processed. So that was kind of the rationale behind the storefront for art and architecture project, like using wood as a bioreceptive substrate. It's a double-edged sword though, because then that material would likely be more prone to biodegradation. So I think that depending on the time frame at which you're designing and like what the lifespan of the structure is that you're imagining, you would want to be able to tune that.
Thank you. Any other questions from the students?
Does microbes have colors?
Do they have colors?
Yeah.
Yes,
Or
some of them produce pigments. Yep.
Oh.
Yep. Some of them produce pigments. Oftentimes those pigments are related to antimicrobial activity. So they secrete these molecules to be able to fight other microbial species, and those molecules happen to also be pigments that respond to the visible light spectrum. You should check out the Fab Textiles group. They've done a lot of really awesome work with microbial pigments and textiles. I think Natasha is on this call. I think that first question was from Natasha.
Oh, nice. Yeah, I'll definitely check that out.
Cool. Then I think if there are no more questions, we can wrap this up. So thank you very much, Elizabeth. It was a super interesting conversation, and I hope that the students got some ideas from it for their own projects and for MDDI. So yeah, thank you very much, and
You're very
we'll see
welcome.
you,
Always a pleasure.
soon.
Yeah.
Thank you.
Bye, guys.
Thank you, Elizabeth.